Okay, so now that we know the structure of DNA and we've learned a bit about DNA replication, okay, now we're going to talk about new things, right? So DNA replication, we hadn't talked about how it happens yet. I mean, other than this last video, right? But we know when it happens, right? So it happens during interphase. Cool. Okay. So before we can talk about the other activities of DNA, we need to talk about RNA for a second. Okay, so the other sort of main category, at least as far as we're concerned, the other main category of nucleic acid that we're going to be talking about is RNA. And RNA stands for ribonucleic acid instead of deoxyribonucleic acid. Okay, RNA. How is RNA different from DNA? There are several ways. One significant way is that RNA is single-stranded. So DNA, recall, spends most of its time in a double helix, right? RNA does not. RNA is typically single-stranded. So that's one difference. Another important difference is that the actual nucleotides are a little bit different. So one way that's different is in the type of sugar. So the phosphate is the same. That's the same. The sugar, it also has a sugar group, but in this case, instead of it being deoxyribose, guess what? It's ribose. So it's a different sugar. Okay, so this is one difference. Here's another difference. And then the third difference is that RNA, because it is also nucleic acid, the nucleotides of RNA also have a nitrogenous base. But you'll notice here that it says can be A, G, C, or U. We didn't talk about U before, okay? So U stands for uracil. Instead of thymine, okay? So remember the T was thymine. And so uracil is found in RNA. Thymine is found in DNA. The other three are found in both, okay? But that's one difference, okay? How do you deal with this? Well, I mean, the easiest thing to do is just like sub it out, right? So what were our base pairing rules for DNA? Let's take a minute to think about that. So which two went together? C went with G, right? Or G went with C. Same difference, doesn't matter which one's first. Okay, so that we can still do that in RNA because we have C's and G's. We don't have T's anymore though, so who is going to be A's partner? Who's going to pair up with A? You. So you just swap it out essentially, okay? So uracil is the substitute, right, for thymine in the case of RNA, okay? So those are the main ways that RNA is different. Now, of course, what RNA does is different as well. We'll get to that, okay? But those are the primary differences between DNA and RNA. That interruption was just long enough for me to completely lose my train of thought. Okay, so I think we got RNA, I think we talked about the differences. If not, it'll come up as we go. Okay, so DNA replication happens specifically so that you can have new DNA when you make new cells, okay? But that is not the only time that DNA does things, because as you may recall, the other thing that DNA is responsible is for, making stuff, right? So this is sometimes referred to as the central dogma of biology, which sounds really like intense, okay? But the idea is this, all right? You have DNA in your chromosomes, right, which are found in your cells. That's what all of this part is about, okay? DNA directs the production of RNA. So DNA essentially is used as a template to make RNA. And then RNA is used as a template to make protein. And ultimately, those proteins are what is responsible for all kinds of different traits. And so this lists some of the ones that we talked about in the genetics chapter, right? So whether or not you have a widow's peak, right? Whether or not you have freckles, you know, what kind of blood type you have. Yeah. So this is what we're going to talk about next. And we're going to break it up into 2 videos because I want it to be very clear what the separation is between those 2 activities. Okay. Here's another little illustration of the central dogma. So this is the process, right? This shows you where all of that is happening. Okay. So the purple thing is the nucleus, right? DNA is found inside the nucleus. It never leaves the nucleus except during cell division when the nucleus breaks apart, but whatever. Okay. So DNA stays in there. Now, the nucleus has pores or openings in the envelope. Okay. DNA can't fit through those pores. So why are they there? Well, there's a couple of reasons. One reason is that RNA can pass through the pores, and also the components of ribosomes as well, because ribosomes, excuse me, are actually made inside of the nucleus. Now, this drawing is not to scale. You're looking at this ribosome right here, and you're like, that can't fit through there. What are you talking about, lady? So this is not to scale, all right? But the little parts of ribosomes, the little components that make up a ribosome, can also fit through these little pores because ribosomes are built inside the nucleus. So another good thing to think about is, what do ribosomes do again? When did we learn about that? We learned about that in cells, cell chapter, which is chapter 3. Okay. All right. So these 2 processes, right? This process here, DNA directing the production of RNA, and then RNA directing the production of protein, are called transcription and translation. Okay. Transcription is basically the process in a general sense, like in a general, like English language sense. Transcription means you're taking something from one form of a language and putting it in a different form of the language. So that thing, that writing that's on the side of the screen, if you open up this video, is the transcript of what I'm saying. Okay, so transcription is happening. My words, my auditory words, are being converted into a different form of the same language, a written form. Okay, that's what's happening in transcription inside cells. Transcription occurs, notice, look at that, inside the nucleus. Okay, and essentially what transcription is we're taking one form of nucleotide language, which is DNA, to make a different form of nucleotide language, which is RNA. Okay, that's transcription. So we're going to go through and we're going to talk about the parts of transcription in a minute. Translation, once again, let's talk about sort of just the general usage of the term. Translation typically means you're converting from one language to another language, right? So, you know, if somebody wanted to, you know, was watching my videos and they didn't speak English, right? They speak Japanese. I'm just going to pick something, right? So one thing they could do is they could take that transcript, right, that comes up, they could take that file and they could put it through some sort of translator program, like use Google Translate, right, to turn it into Japanese. I can talk. Okay, so that is translation, going from one language to a different language, a completely different language. Okay, so in the case of the cell, the RNA is skinny enough that it leaves the nucleus, right? So the RNA sneaks out of those pores, right? And then translation occurs outside of the nucleus, specifically on ribosomes. Now, if you think back to the cell chapter, you might recall sometimes ribosomes are just floating around in the cytoplasm, like, you know, free. And sometimes they're attached to endoplasmic reticulum. That's right. Okay. But translation occurs on ribosomes. Okay. All right. And so in this case, what we're translating is the language of nucleotides, right? So the RNA to the language of proteins, totally different type of macromolecule, totally different language. Okay. All right. So that's kind of like the basics of the central dogma. 2 steps, transcription and translation, and a little bit of a reminder for how you remember which one is which. Okay. All right. So the rest of this video is going to be about transcription. And then there's going to be a separate video about translation so that we can kind of keep them. They both start with trans and end with shun. So it's really easy to get them confused. So I'm trying to physically separate the videos so that you, you know, can look at them separately. Okay. All right. So transcription. Here's a picture of transcription. All right. Now, I don't really care if you know the names of the steps, right? So initiation, elongation, termination. Basically, it starts, it goes, it stops. That's all that means. Okay. Ooh, there's an orange blob. So the blue double helix that has kind of a green area and a red area, that represents our DNA. Okay. And so this is the DNA of some gene. Okay. A segment of DNA on a chromosome. And this orange blob represents something called RNA polymerase. Let's think about that for a second. We know what DNA polymerase does. What do you think RNA polymerase does? So DNA polymerase was responsible for building, it's an enzyme that builds DNA polymers. So RNA polymerase is an enzyme that builds RNA polymers. My goodness. Right? Okay. So how does this go down? Well, RNA polymerase is floating around. Remember, this is all happening inside the nucleus, right? Transcription, in fact, inside nucleus. Maybe what we'll do is we'll use this slide as a running like list of important key points. Okay. So this is happening inside the nucleus. So what happens is that RNA polymerase binds to the DNA at a specific site called the promoter. I don't really care that you know that name necessarily, but the idea is there's a spot that's like, hey, RNA polymerase bind here. Okay, so it binds to a specific section. Okay, then what the RNA polymerase does is it behaves somewhat similarly to DNA polymerase in the sense that it pulls the DNA apart and it's building something. But the something that it's building is RNA instead of DNA. Okay. And it kind of travels down the strand of DNA, down the double helix of DNA, until it hits an area, it's shown here in red, right? Called the terminator. Termination, just a fancy name for stop, right? So starts at the promoter, ends at the terminator, starts at the beginning, ends at the end. Okay. And the terminator basically, that area, the RNA polymerase can't bind to it anymore. So it kicks it off and then everything's separated. Okay. So it's not super complicated, right? Oh, okay. So there are videos. There's a bunch of videos that I linked, and there's other videos that are in your homework. So you're going to see, if you're looking at this and you're like, I really need to see a video of this, thank you very much. There are videos all over the place in the page that has these lectures and also in on YouTube. And I linked a bunch. Okay. All right. Whew, so here we go. This is what the beginning looks like, right? So here's our R, or I'm already screwing it up, right? This blue line represents our double helix of DNA, okay? And so our RNA polymerase, the orange blob, jumps on at the section called the promoter. It can't jump on just anywhere. It has to jump on in that specific spot, okay? Now, going from real life looking to fake looking, back to real life looking, okay? These fake images are not in your book, but I like them because they're a little less, it's a little easier to like focus, I think, I hope, or they're confusing. Either way, this image and this image essentially are the same, right? If I flip back and forth, they're essentially the same, right? So if you like this better, go with this. If you like this better, go with that. Okay. So this is showing sort of, we're in the middle of, so this is not at the very beginning where the promoter happens, right? This is after that. Okay. So what the RNA polymerase does is it pulls the double helix apart. So it pulls the strands apart and it brings in RNA nucleotides that match up with one side of the DNA. One of them acts as a template, the other side is just nothing's happening there. Okay? And, you know, because of base pairing rules that you already learned, right? If our DNA is T, A, G, G, T, T, da, 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 da. Okay? What matches up with T? Oh, that's A. What matches up with A? Oh, that's T. Wait a minute. Remember that RNA doesn't have T's. So if we're talking about making RNA, it's going to be a U instead of a T. Okay? What goes with G? C. What goes with G? C. What goes with T? A. What? Yes? Okay, you'll have opportunities to practice this, right? And that strand of RNA grows and grows and grows as the RNA polymerase travels down the length of that double helix of DNA until it hits the terminator, okay? So once again, they're trying to show you a more realistic looking picture, right? So the orange blob is the DNA polymerase, RNA polymerase. See, there I go, being confusing, right? And remember that the double helix of DNA is pulled apart. One part is not being transcribed, it's being ignored, right? One side is being transcribed. Okay. And so these individual little RNA nucleotides are coming in to where they match up according to base pairing, right? And as the RNA polymerase is scooting down that double of helix of DNA, it pulls it apart long enough to build that RNA, which is growing longer and longer and longer. And then as it's moving down the DNA just sticks back together. Okay. That's cool. Yeah. So here's the RNA polymerase scooting along that double helix of DNA. And coming out the back end of the RNA polymerase is the growing strand of RNA. And there's this other enzyme here that is actually basically pinching out and cutting out, we call that splicing, cutting out sections of that RNA that we call introns. Okay. All right. Like I said, there's videos for all of this. So if you're like, I want to see this, I want to see it happen, I want to see an animation of this, go back and look at all those videos or look at them before you do this. I don't know, whatever. Okay. All right. So let's talk about that coding versus non-coding splicing intron business. Okay. As it turns out, much of the DNA that is on your chromosomes, right? This is in your chromosomes, makes up your chromosomes, is not used directly to make a protein, believe it or not. People used to call it junk DNA. Some of that might be kind of true. Some of it is just like, just because we don't know what it does doesn't mean it's junk. Okay. But there's a lot of stuff that's in there that actually gets cut out. Okay. And what we call that stuff that gets cut out is we call it a internal non-coding region. Okay. So here's a gene, here's little bits. Right? So most of the regions that don't code for a protein are in between genes, right? So like this is not within a gene, okay? But there are some areas that don't code, right? That are inside of the gene. And when they are found inside of the gene, we call them internal non-coding regions. The word we use to abbreviate that is intron. Okay, so intron means internal non-coding region. Okay. All right. So exons are the pieces that code. Okay, so what we're looking at in this image, once again, this is not from your book. What we're looking at in this image is we're looking at something called RNA processing. Come on, pen. RNA processing. Okay, so here's our DNA at the top, right? Exon, intron. Okay, it's transcribed, right? Here's transcription happening. In addition to all of this, what's happening next, another thing that happens in RNA processing is adding a cap and a tail. Okay, and they're represented there in yellow. Essentially, the way that I like to think of the cap and the tail is they're like little bumpers that protect the RNA, because the RNA has to go from inside of the nucleus out into the cytoplasm, and there's all kinds of, you know, things it could run into, right? So they're like little bumpers that like protect the actual message, right? Protect the code, if you will. Okay, so a cap and a tail are added, and introns are removed, right? So there's an intron, here's another intron. And so they are quite literally cut out by little enzymes that look like this, little splicing enzymes. So those little splicing enzymes find the sequence where they need to cut that piece out, and they cut it out, right? They literally like merge the other two ends and cut out the junk that's in between. Okay. Okay. Now we've got to then stick the exons back together, right? So probably more ligase involved. Sticking together. Okay. Then what we've produced is something that we call mRNA, and that stands for messenger RNA. Okay. So what happened in DNA processing? Well, the first thing that happened, well, one thing that happened is the addition of the cap and the tail, right? And then the introns removed and the exons were spliced together, were stuck together, okay? And then that original RNA transcript now becomes, we call it something called mRNA, and that stands for messenger RNA. That messenger RNA then leaves through one of those nuclear pores. Hey, look, there it's leaving. Right? So look, oh, look at that little yellow bumper. There's the cap and there's the tail, right? And it's called mRNA by the time it leaves the nucleus. We're calling it mRNA. Yeah? Okay, that's transcription. So we'll stop here and then we'll pick up and talk about translation. So, what else do we want to say? So it happens inside of the nucleus, okay? RNA polymerase. Pulls DNA apart, builds RNA. Okay. Then the RNA is processed, right? Those were the last slide we just looked at. And leaves the nucleus. Okay, so that's like bare bones of what transcription does. Okay, all right, we'll stop here, and the next video will be translation.